Okay, so welcome back to this next video in which we are discussing cystic fibrosis and cystic fibrosis medications. Okay, so we're currently in the process of discussing the pathology of cystic fibrosis. So we've discussed the cystic fibrosis transmembrane conductance regulator and how it's this ATP-gated chloride channel. We've then discussed the different mutations that can arise in the cystic fibrosis transmembrane conductance regulator, and we've divided those into 5 different classes. Okay, we're now discussing why loss of function in the cystic fibrosis transmembrane conductance regulator is going to lead to the symptoms of cystic fibrosis, and we're starting off with the incredibly salty sweat. Okay, so we have discussed the structure then of an eccrine sweat gland. We've discussed how they consist of a sweat duct here, and then the coiled portion, which is the actual sweat gland. And the sweat gland is going to have cells that are cuboidal epithelial cells, which are going to be secreting the primary secretion of the sweat gland, which is a watery solution containing a large amount of sodium and chloride ions. Okay, and it's originally going to be isotonic to the extracellular fluid, so it's going to have the same concentration of solute in it as the extracellular fluid. There is not going to be an osmotic gradient between the extracellular fluid and the primary secretion of the sweat gland. Then what will happen is the primary secretion will be forced up the sweat duct and out onto the surface of the skin. Now, you get to the point in the sweat duct where it becomes very, very watertight, okay, and water now cannot move across this epithelium, okay, and that's the case with the skin as well. Clearly the skin is watertight, okay, otherwise every time you go out in the rain you'd be absorbing rainwater, okay. So once the primary secretion goes up here, okay, water cannot move across that membrane anymore. So what now happens is the cells of the sweat duct, known as the duct cells, and I'll just write that down, the cuboidal epithelial cells that line the sweat duct, those are going to be known as duct cells, okay? These duct cells are going to be removing the solute from the sweat, okay? They're going to be removing sodium and chloride from the sweat, okay, and they're going to be reabsorbing those, which means that the solute concentration in the sweat is going to decrease as it goes up the sweat duct, okay? Now that will make it hypotonic compared to the extracellular fluid, and you might think, well, surely now water will move by osmosis back into the extracellular fluid, but then you need to remember that the sweat duct epithelium is extremely watertight, so it can't move anymore. So that's how we can build up this hypotonic solution here, and then that will go onto the surface of the skin. Now in cystic fibrosis, what's going to happen is the ability of the duct cells to reabsorb sodium chloride is going to be reduced by the loss of function of CFTR. Okay, and that means that your sweat is going to become more salty, because now it's going to have the same salt concentration as the primary secretion down here in the sweat gland. Okay, because now these duct cells haven't resorbed the sodium chloride out of that primary secretion to produce the hypertonic sweat that is normal. Okay, right. So what we now want to do is discuss the mechanism by which these coil cells down in the actual sweat gland, firstly how they secrete the primary secretion, and then we want to see how the duct cells reabsorb the sodium chloride, and we'll see where CFTR is involved and why the primary secretion is not affected in cystic fibrosis. And quite simply, it's because these coil cells do not use CFTR as their chloride channel. There are many other chloride channels. They use different chloride channels to CFTR, whereas the duct cells, those use the CFTR chloride channel, and that's why they're affected in cystic fibrosis. So, let me get another piece of paper and we'll have a look at this then. Okay, right. So, I'm going to draw a coil cell then, and we're going to see how the primary secretion of the isotonic solution to the extracellular fluid is produced. Okay, right. So I'll draw this nice and big so that I'm not struggling for space. Okay, so this is going to represent our coil cell. So this is one of those cells in the coil portion of the sweat gland. Okay, Now, with any epithelial cell, there are two portions of the membrane. There is an apical portion of the membrane, and then there is also a basolateral portion of the membrane. Okay, so the apical portion is the portion that's going to face into the lumen of the sweat gland. Okay, so this is the apical face, and then the basolateral face is the face that doesn't face into the lumen of the sweat gland. Now, these two portions of membrane are physically divided. There are tight junctions which go around the entire circumference of the cell. Okay, so because we're taking a cross-section of the cell, we're only seeing a little section of them, but in reality they're coming out of the page and wrapping around the entire cell and separating these two halves of the cell membrane, basically, the apical membrane and the basolateral membrane. Okay, so these represent tight junctions, and they are so important, because now what it means is that you can put proteins, integral membrane proteins, in the basolateral membrane, and those can float around in the basolateral membrane, but they can't get into the apical membrane because they will come along here and they will hit the tight junction, and that is quite simply a physical obstruction that stops these proteins from being able to move into the apical membrane. So basically, you can put certain integral membrane proteins in the basolateral membrane and different integral membrane proteins in the apical membrane, and they're not just going to end up floating into the other one, basically. Okay, and that's a very key concept to understand about epithelial cells, that the integral membrane proteins in the apical membrane are truly separate from those in the basolateral membrane. Okay, so firstly then, in the basolateral membrane, you are going to have the sodium-potassium ATPases of the cell. Okay, now all cells have sodium-potassium ATPases. They are one of the essential proteins. Okay, but epithelial cells are going to specifically have the sodium-potassium ATPases in the basolateral membrane. Okay, and I should say these coil cells are going to have it there, but most epithelial cells do end up having them in their basolateral membrane. Okay, now what does sodium-potassium ATPases do? They throw 3 sodiums out and they bring 2 potassiums in, and every time they do that they use a molecule of ATP. So ATP will be hydrolyzed down to ADP and inorganic phosphate. Okay, so it's a primary active transporter here. So this is the sodium-potassium ATPase. Now, as I say, all cells have these. Okay, what's special here is that they're all going to be confined to the basolateral membrane in these coil cells, and generally in most epithelial cells. Okay, I think probably in all epithelial cells I've come across. Okay, and certainly in all the epithelial cells we're going to see in this video. Okay, so the sodium-potassium ATPases are on the basolateral membrane. Now, what does the sodium-potassium ATPase do? It produces the sodium and potassium gradients across the cell membrane. Okay, so sodium, remember, is usually much higher in the extracellular fluid than in the cytoplasm. Usually, sodium concentration extracellularly, I'm wondering where to write this because I'm going to need more space here to put other proteins on. Okay, I'll put it up here. Sodium concentration extracellularly is usually around 145 millimolar. Okay, whilst intracellularly it's generally around 12 millimolar. Okay, that's sodium. So again, we can see that the sodium-potassium pump is moving sodium out of the cell, and that's responsible for maintaining this concentration gradient across the cell membrane. Okay, so potassium then, what's the potassium gradient across the cell membrane? Well, the potassium gradient across the cell membrane is generally around 155 millimolar in the cytoplasm of the cell and 4 millimolar extracellularly. Okay, and that's because you're bringing potassium into the cell, so it's going to end up higher in the cell. Now you might be wondering, well, why on earth is the potassium gradient so much more stronger than the sodium gradient? Okay, you also have to take into account other factors when you actually work out what these concentrations are going to end up as. Okay, so it's not just the sodium-potassium pump that is establishing those. Okay, but they certainly help. Right, so the sodium-potassium pump produces these sodium gradients and this potassium gradient across the cell membrane. Okay, right. So what then is going to happen is you're going to have another protein on this basolateral membrane here. Okay, and this other protein is what's known as the NKCC. Okay, now what does this stand for? This stands for sodium. That's what the N is. Remember, the periodic table symbol for sodium is Na. Okay, so it's using the periodic table symbol there, N. Okay, then potassium, that's the K. Okay, again using the periodic table symbol. Then the C is for chloride. Okay, and then the final C is for co-transporter. So that's what the NKCC stands for. It's the sodium potassium chloride co-transporter. Okay, and I think I'll colour in the sodium potassium chloride co-transporter in vivid purple here. So this is the sodium potassium chloride co-transporter. So, the sodium potassium chloride co-transporter is going to use the sodium gradient across the cell membrane to move potassium and chloride into the cell. Okay, so what you're going to do is you're going to bring in one sodium into the cell. Okay, now the sodium will be coming down its electrochemical gradient. Okay, now what does that mean? Well, basically, it's not just about the concentration gradient. When you consider whether something wants to move in or out of the cell, you have to consider more than just the concentration gradient across the cell membrane. You also have to consider the electrical gradient across the cell membrane. Now, usually across the cell membrane, the electrical gradient is around negative 65 millivolts. Now, I'm wondering where to put this. Squeeze it in here. Negative 65 millivolts. Now what that means is that the electrical potential intracellularly, whatever it is 65 millivolts lower than the electrical potential extracellularly. Okay? Now, sodium and potassium are both positively charged ions. Okay? Now, positively charged ions want to go where the electrical potential is lower. Okay? Which is currently the cytoplasm. Okay? So the electrical gradient is favouring the movement of sodium in and potassium in. Okay, now clearly sodium's concentration gradient also favours the movement of sodium in. So when you bring sodium into the cell, that releases energy not just because you're bringing it down the electrical gradient, but because you're also bringing it down the concentration gradient. So we say that sodium's electrochemical gradient favours its movement into the cell. Okay? Right. Now, potassium is also going to come into the cell. Now, potassium's electrochemical gradient is more complicated because the chemical gradient, the concentration gradient, favours the movement of potassium out, but the electrical gradient is favouring the movement of potassium in. So this is more of a puzzle. Which one wins? Well, to decide which one wins, you need a lot of maths. Okay, but I'm just going to tell you the answer. The chemical gradient wins at negative 65 millivolts. This is not powerful enough to beat this concentration gradient. Okay, so potassium's electrochemical gradient overall favours its movement out. However, it's very, very small because the electrical gradient is fighting the concentration gradient. So basically, potassium would rather move out, but if you do move it out, you would only release a tiny little amount of energy, and therefore when you move it in, you only have to put in a tiny little amount of energy. So it's almost negligible, basically, the energy that you have to put in to move that potassium in. Okay, so the thing to focus on here is that sodium is coming in, down its concentration gradient, down its electrical gradient, so it's releasing a huge amount of energy. Okay, then you've got the potassium coming too, and then you're also going to bring in 2 chloride anions here as well into the cell. Okay, and they will be brought in on the back of the energy that the sodium is releasing when it comes down its electrochemical gradient. Okay, so you're bringing chloride anions from the basolateral extracellular fluid into the cytoplasm of the cell. Okay, that means that chloride anions are going to build up in the cytoplasm of the cell. They are then going to move out of the cytoplasm of the cell into the extracellular fluid on the apical side, i.e. into the fluid in the lumen of the sweat gland. Okay, so we're dumping chloride anions basically into this space here. Okay, that's what I mean by the lumen. Okay, so the apical faces of these coil cells are facing into the lumen of the sweat gland. Okay, so the chloride anions are going to move out of the cytoplasm and into the lumen of the sweat gland. Okay, through a chloride channel here. Now, this is a coil cell. I said that cystic fibrosis was not going to affect the coil cells. So hazard a guess as to whether this chloride channel is a CFTR chloride channel. The answer is, of course, no. This is a chloride channel, but it is not CFTR. Okay, so I will just put there not CFTR. Okay, now, these cells may have some CFTR on their apical membrane, but it doesn't seem that it is the essential one for the function of these cells, okay? So it seems that there are other chloride channels that are more important than CFTR. So I'm not saying that there is absolutely no CFTR in the apical membrane. There may well be, but the main one that is responsible for the function of the coil cells is not CFTR, okay? And that's why these coil cells are not going to be hugely affected by cystic fibrosis. Okay, right, so that's where the chloride anions are going, okay? The sodium ion will of course be pumped out by the sodium-potassium pump. The potassium ion that you're bringing in, that will go out for a potassium channel here, okay, which is on the basolateral side of the membrane. Okay, right, so potassium will then go back out. Now, if we then look at what we have overall done here, okay, what we have overall done, this potassium that you brought in will end up going back out. Okay, so there's no net movement there, it's just going round and round in a cycle. The sodium will also go round and round in a cycle. It will come in through here and then be pumped out by sodium-potassium ATPase. But the chloride anions, they are not going round in a cycle. They are being moved from the basolateral extracellular fluid into the apical extracellular fluid. Therefore, you are moving negative charge from here to here. That is not sustainable, okay? That is going to set up electrical potential differences between here and here. And that's going to attract positively charged ions. So what's going to happen is sodium ions in the extracellular fluid on the basolateral side are going to feel attraction towards the chloride anions that are now in the extracellular fluid on the apical side. Okay, and I'll colour in this chloride anion over here. So what's going to happen is sodium ions are now going to move into the extracellular fluid on the apical side. Okay, now they're not going to go across the cell. They are going to go between the gaps, okay, between the cells. Okay, so if I draw another cell here, okay, here is another coil cell beginning here. I won't draw the entire thing. Okay, this is the tight junction. Now, the tight junctions are not so tight that they don't allow sodium ions to move through. Remember what the point of the tight junctions was. It was to separate the basolateral side of the membrane from the apical side of the membrane. It was to stop great big integral membrane protein complexes, like potassium channels, from moving from here to here. Okay? That's pretty easy to do, okay? That you just need to put an obstruction here and it will stop something massive like that moving through. Okay, the extracellular portion will get stuck, basically. It won't be able to get past this. A tiny little atom, like a sodium ion, is still going to be able to get through the tight junctions. They're not that tight, basically. They are good, but they're not that good. So sodium ions are going to move across the tight junctions, and this route is known as the paracellular route. Okay, so we say that sodium is going to move paracellularly. Okay, so the sodium ions are going to move between the cells from the fluid of the extracellular fluid on the basolateral side to the extracellular fluid on the apical side. Okay, so now you've overall moved sodium chloride from the basolateral extracellular fluid to the apical extracellular fluid. Finally, what's going to happen is water molecules are then going to move by osmosis, okay, because you're making the solute concentration here much higher and reducing the solute concentration here, so water is then going to move by osmosis, and again it's going to go via the paracellular route. Okay, so that's how you secrete an isotonic solution. Okay, of course it's going to be isotonic because it's going to get to the point where it's in osmotic equilibrium with the extracellular fluid on the basolateral side. That's the whole point of how the water moves. The water moves by osmosis, and therefore it can only get to an isotonic state because that's what osmosis is all about. It's about making this side and this side isotonic. Okay, so then you'll end up with a salty solution that is isotonic to the extracellular fluid on the basolateral side. Okay, right, so that's how the primary secretion is made by these coil cells of the sweat glands. Okay, now note the absence of CFTR. Okay, it is not important for this primary secretion by the coil cells. If we knock out CFTR, as we do in cystic fibrosis, let's say we've got a double delta F508 mutation, okay? So a delta F508 mutation in my maternal gene and a delta F508 mutation in my paternal gene. You've got no functional CFTR now at these apical membranes, okay? These coil cells will still function because this chloride channel is not CFTR, okay? So you still get the primary secretion produced. So the volume of sweat that you produce is not harmed at all in cystic fibrosis. Okay, what's going to go wrong now is what's supposed to happen in the duct cells of the sweat gland. Okay, so what we're now going to move on to is moving away from these coil cells here, and we're going to go up and look at these duct cells here. We're going to look at how they reabsorb sodium chloride from the solution that's passing through the lumen here, okay, and we're going to see how that's dependent on the cystic fibrosis transmembrane conductance regulator. Okay, whoops. So I'm now going to draw a duct cell, and we're going to see the mechanism by which it reabsorbs sodium chloride from the duct. Okay, so same protocol. I'm going to draw a big square blob of a cell here. Okay, this is going to be the apical side here, which will be facing into the lumen of the duct. Okay, and this is going to be the basolateral side. Whoops, let me just put an O, squeeze a O in there. This is going to be the basolateral side over here, which will be facing basolateral extracellular fluid and of course the basement membrane, which will be underneath. Okay, and we've now got tight junctions that are so tight that they are not going to allow water in between, so you're going to have much more connections between these cells now. Okay, stronger junctions that are going to make this truly watertight. Okay, right, so let's now discuss how these cells then are going to absorb the sodium chloride from the solution that is passing through the duct. Okay, so the first thing then to say is that once again you have sodium-potassium pumps on the basolateral membrane here. Okay, so I'll just colour this in the same colour. So in green here, this is a sodium-potassium pump. Okay, so we'll be chucking 3 sodiums out and 2 potassiums in, and that's producing the concentration gradient of sodium and potassium across the cell membrane. Okay, so note again that it's located in the basolateral portion of the membrane of these duct cells. Okay, right, then we're going to have a sodium channel on the apical membrane which is going to be absorbing sodium from the lumen. Okay, so sodium is going to be much more concentrated in the sweat fluid that is moving through the lumen of the duct than it is in the cytoplasm of these epithelial cells. Okay, so I'll colour in this sodium ion here in green. So now we're going to have a sodium channel in the apical membrane of these epithelial cells, which is going to be allowing sodium ions to move from the extracellular fluid on the apical side, i.e. the sweat, back into the cell, and this is how we're going to be removing sodium ions from the sweat solution. Okay, so what is the name of this sodium channel? Well, it's a very famous sodium channel known as ENaC. Okay, now what does this stand for? This stands for the epithelial, okay, that's the E, then the Na is for sodium, okay, channel is the C, so the epithelial sodium channel, a perfectly reasonable name. Okay, so I'll colour it in now in vivid purple. So this is ENaC here. Okay, right. So that's how you're going to bring sodium back into the cell. How are you going to then bring chloride back into the cell? Well, normally chloride concentration gradient again favours its movement into the cell. Okay, so usually you have much lower chloride concentrations inside the cell than you do outside the cell. Okay, so you have a concentration gradient that's favouring the movement of chloride into the cell. Okay. However, of course, the electrical gradient across the cell membrane is going to favour the movement of chloride out of the cell, because the electrical potential inside the cell is 65 millivolts lower than the electrical potential outside the cell. Chloride ions have a negative charge and are therefore going to want to go out of the cell where the electrical potential is higher. Okay, so there isn't really that much of an electrochemical gradient at all for chloride across the cell membrane, okay, because the electrical gradient says go out and the concentration gradient generally says go in. However, it's going to now want to follow the sodium ions, because remember, you're bringing positive charge into the cytoplasm of the cell. So what's going to happen is the chloride anions are going to follow the positively charged sodium ions, so that you're not overall bringing charge across this cell membrane. Okay, if you were just bringing the sodium, you'd be making this more and more positive, okay, and you'd be disturbing the electrical potential difference across here. Okay, the chloride is going to be attracted by the sodium and therefore follow it and neutralize that change in electrical potential. Okay, so chloride anions then are going to move from the apical extracellular fluid into the cytoplasm of the cell here, okay, with the sodium ions. And I think I'll colour in the chloride anion as well. Now, what is this chloride channel that is on the apical membrane of this duct cell? Well, this then finally is the cystic fibrosis transmembrane conductance regulator. Okay, so this is why cystic fibrosis is going to affect these duct cells. But we'll see the complete picture before we come back to what cystic fibrosis is going to do to these duct cells. Okay, so you've now reabsorbed sodium chloride into the cytoplasm of the cell. The sodium then is going to get chucked out of the apical membrane, of the basolateral membrane, by the sodium-potassium pumps. Okay, so this sodium is then going to get eventually pumped out by sodium-potassium pumps into the basolateral fluid. Okay, so eventually the sodium will end up being moved from here to here. That means again that you will move the sodium from the cytoplasm to the basolateral extracellular fluid, and again that means that you've moved sodium across this membrane. So what will then happen is the chloride will want to follow the sodium. Okay, so again you have a chloride channel on the basolateral membrane through which chloride anions can move. And it's still contentious as to whether this chloride channel is CFTR as well, okay, or whether it's some other chloride channel. Okay, so I'll just call it a chloride channel. It may well be CFTR as well, okay, but CFTR is famously in the apical membranes of these duct cells. Whether it's also important in the basolateral side of these duct cells is still controversial, okay, but some chloride channel exists in the basolateral membrane that is allowing the chloride to move into the basolateral extracellular fluid. Okay, so to paint the full picture here, what is happening is we are moving sodium chloride from the apical extracellular fluid to the basolateral extracellular fluid, okay? And the driving force for this is that sodium's electrochemical gradient always favours its movement into the cytoplasm of the cell. Okay, so the sodium moves through the ENaC channel into the cytoplasm of the cell. Chloride anions will then also follow the sodium ions and move into the cytoplasm of the cell. The sodium will then get chucked out at the basolateral side of the membrane into the basolateral extracellular fluid, and the chloride will follow into the basolateral extracellular fluid. Okay, and that is how we are moving sodium chloride from the apical extracellular fluid into the basolateral extracellular fluid. So we're removing salt from the sweat. Okay, if I get my picture of the sweat gland back again, this is where we're working, and we are removing sodium chloride from the lumen of the sweat duct into the basolateral extracellular fluid. Okay, now that is going to decrease the solute concentration in the sweat. Okay, and it's going to, of course, increase the solute concentration in the basolateral extracellular fluid. So you should think that water would move from here to here by osmosis, but then comes the important statement that these cells are watertight. The junctions between the two cells are very watertight, so the water cannot move, basically. The water is trapped in the lumen of the sweat duct, and that makes your sweat hypotonic, okay, to the extracellular fluid. It has a lower solute concentration than the extracellular fluid of your body. This hypertonic sweat will then be secreted onto the surface of the skin and will then cool you down. Okay, right. So, cystic fibrosis then. What happens in cystic fibrosis? In cystic fibrosis, the coil cells are unaffected. They produce the primary secretion just fine. The duct cells, however, are affected. This is lost in the duct cells, okay? And if the CFTR is lost in the duct cells, then the whole thing is going to stop, because even though the ENaC is still functional in the duct cells, if you're bringing in the sodium into the cytoplasm of these duct cells, you're going to very quickly create an electrical potential difference across this cell membrane that is going to oppose the movement of further sodium in. Okay, you can't just bring positive charge from here to here. Okay, you have to bring a charged ion that will counterbalance the movement of positive charge. If you don't, then the sweat will just become more and more negatively charged. Okay, but as you move more and more sodium out. Okay, and that's not sustainable. Okay, that will build up very quickly and it will stop any further movement of sodium ions in. Okay, so it is essential that you bring the chloride too if you're going to reabsorb sodium chloride. You can't just reabsorb the sodium and leave the chloride, because, as I say, you'll create sweat which is going to be negatively charged, okay, which isn't doable. At least you can't do it to any great extent. Okay, so what's going to happen is sodium absorption is going to be stopped very quickly, basically. Okay, so going to stop reabsorbing sodium chloride by the duct cells, okay, and that means the sodium chloride will remain in the sweat, it will be secreted onto the surface of the skin, and you'll therefore end up with very salty sweat, sweat that has a very high concentration of sodium chloride within it. Okay, right, so that now is the explanation of why cystic fibrosis results in salty sweat complete. Okay, so that's our first symptom of cystic fibrosis, and you might think, goodness gracious, we spent so long just on this one symptom. Okay, it will help us understand all the other symptoms. Okay, the lungs and the pancreas and the vas deferens, they all use these same pathways that we have just been talking about. Okay, so now that we've done this, it sets the stage for understanding the pulmonary symptoms, the pancreatic symptoms, the intestinal symptoms, and the male reproductive tract symptoms. Okay, right. So I think we will call it there for this video, and in the next video what we will do is we will move on to the respiratory symptoms. We will start by looking at a little bit of the anatomy and histology of the bronchial tree, because that's important so that we can understand what the mucosal glands are. Okay, and then we will look at the actual secretion by these submucosal glands, and then we'll see how cystic fibrosis is going to impair their function and how that's going to lead to recurrent pulmonary infections.